Folks, the talk is Sexual Transmission of American Trypanosomiasis in Humans, Insights into Evolution and Pathology in Chagas Disease. The protozoa and Trypanosoma cruzi long-lasting asymptomatic infections carry a new potential pandemic route for the Chagas parasites. On the one hand, the sexually transmitted T. cruzi infections recognized by the parasite nuclear DNA PCR in the absence of the parasite antibody. On the other, the T. cruzi kinetoplast DNA PCR assessments reveal the mitochondrial minicircle sequence transfer into the human genome. The vertical transfer of the kDNA sequence associates biodiversity and Chagas disease. The Trypanosoma cruzi undergoes life cycle in vertebrate hosts, somatic and germline cells as well. Clumps of mastigotes and trypomastigotes present in the lumen of a seminiferous tubule and the dividing mastigotes in the gonial cells. This figure shows the Trypanosoma cruzi life cycle in the semen of an infected mouse. Amastigotes and trypomastigotes A and B, dividing epimastigotes C and D. The blood trypomastigotes and the intracellular dividing amastigotes show the nucleus and kinetoplast, DNA-containing organelles. The mitochondrial kinetoplast DNA is a disc-shaped network comprising maxicircles and minicircles and decatenation by the topoisomerase II enzyme. The average T. cruzi minicircle sequence encloses 4 230 base pair variable sequence blocks interspaced by 122 base pair conserved blocks. The conserved sequence blocks, CSBs, retaining 35 nucleotide repeats like short repeat senes into the transposable long interspersed nuclear elements line 1 in the human genome. The Trypanosoma cruzi infections transmitted by the insect vector in Latin America hinterland. The T. cruzi infections in the cities are not bug transmitted. The population migration spread the T. cruzi infection to the northern hemisphere. Congenital and sexual transmission are revealed synonymous. The asymptomatic T. cruzi infection in humans usually lasts for life. In this regard, the American trypanosomiasis comprises the silent T. cruzi infections. The clinically manifested cases showing dilated cardiomyopathy, megacolon and megaesophagus, and neuroendocrine syndromes are named Chagas disease. The acute Chagas disease is rarely seen. Average 5 cases reported yearly in the last 30 years. The Chagas heart disease shows increased heart silhouette, cardiomegaly, and target cells rejection by the immune lymphocytes. The mega syndromes show enlarged and thickened walls of the esophagus and of the colon, showing parasympathetic ganglionitis and lysis of the neurons. A short-run cross-sectional family-based research protocol designed to find out the patterns of the T. cruzi infections in the study populations of 2 counties in the Amazon. The methods used to diagnose of the T. cruzi infections confirmed by the demonstration of the parasite, by the indirect antibody assays, and by the amplification of the protozoan conserved telomere sequence in the host's blood cell DNA. The nucleic acids tests carried out with the constructs of short sequences for the PCR amplification of the cruzi nuclear DNA, nDNA, and of the kinetoplast DNA, kDNA. Notes the TCZ12' anneal at both extremities of the 188 nuclear probe. The specificity of the immune diagnosis of the cruzi infections revealed the parasite phenotype by the specific antibody, the archetype cruzi Berenice stock, and the wild type EC1 to EC1 for isolates from family subjects with the parasite demonstrated in the peripheral blood, showing green silhouette by the fluorescent label IgG antibody. The specificity of the immune assays showed by the negative antibody recognition of the kinetoplastid protozoan Leishmania brasiliensis. The cruzi genotype revealed by the PCR amplified parasite nuclear DNA and kDNA sequence confirmed by clone and sequence of amplicons. Notes that the 83-109, 76% family subjects yielded the nDNA PCR positive test, whereas 101-109, 92.6% individuals had the kDNA PCR positive test. This result suggests that the cruzi kDNA sequence integrates in the human genome. This is a hypothesis to be considered. The hiridograms depict patterns of vertical transmission of the Trypanosoma cruzi infections and of the kDNA minicircle sequence. The schematic representation of the results of the antibody and of the nucleic acids assays show variegated assortments of the vertical transmission of the cruzi infections and of the kDNA minicircle sequences alone. 7 patterns of the cruzi sexual transmission depicted. 1. Father, kDNA PCR and nDNA PCR positive. Mother, antibody positive, nDNA PCR and kDNA PCR positive, cases 26 and 27. 2. Parents, nDNA and kDNA PCR positive, cases 28 and 29. 3. Father, all assays negative. Mother, positive nDNA PCR and kDNA PCR. 4. Father, positive DNA and kDNA PCR. Mother, uninfected in all the assays. Cases 36 and 37. 5. Father, kDNA PCR. He is uninfected. Mother, cruzi infected, positive, all assays, 61 and 62. Pattern 6. Father, nDNA PCR and kDNA PCR positive. Mother, uninfected. kDNA PCR negative. kDNA PCR positive, 63 and 64. 7. Parents, cruzi in the blood and all assays positive, cases 89 and 90. Discrepancies among the results of the assays used to diagnose the Trypanosoma cruzi infections. The schematic representation show discrepancies among the results obtained with methods to diagnose the Trypanosoma cruzi infections. The control groups 1 and 2 are respectively negative and positive. Group 3, 28.4 positive, yielded all exams positive. Group 4, 47.6%, with NGNA and kDNA positive, and antibody negative. Group 5, 16.3%, kDNA positive alone. Group 6, 7.3%, all negative. The immune tolerance is confirmed in the chickens hatched from T. cruzi inoculated eggs. The immune tolerance observed in the aves refractory to the T. cruzi infections. Group 1, chicks hatched from the T. cruzi inoculated eggs. Group 2, more controlled chickens received culture medium. The chickens of both groups were immunized with 10 times 10 to the 6 formalin-killed parasites. The group 1 chickens showed immune tolerance in the absence of the specific antibody. The mock control chickens showed immune competence in the T. cruzi antibody. The immune tolerance explained the discrepancies among results of immunofluorescence and ELISA and the NGNA PCR assays. Graphic representation of the immune tolerance in the chicken model system. The T. cruzi antibody titers obtained in controlled chickens immunized with formalin-killed flagellates are significantly different from those of chicks hatched from T. cruzi infected eggs. The PCRs revealed the T. cruzi infections in the semen of 19 adults from 4 study families. The instills of semen aliquots into the vagina or the injection in the peritoneal cavity rendered the T. cruzi mastigos nests in the heart, skeletal muscle, and in the vas deferens of mice. Sexual transmission of Trypanosoma cruzi in mice during intercourse. The breeding of T. cruzi infected male and female mice transmitted the infection to naive mates. The breeding of F1 mice with naive mates rendered the T. cruzi infection to 52% of the F2 progeny. The F2 mice tissue sections showed the parasitic nests in the interstitial of seminiferous tube, in gonioblasts, and in clumps of parasitic forms in the lumen of the tuberous. The infectivity of the Trypanosoma cruzi to the embryo cells in vitro. The T. cruzi infection of a morula stage tissue culture revealed the embryo cells swarm the mastigotes and release of trypamastigotes in the external milieu. Figure 24 shows the early chicken embryo innate immunity eradication of the Trypanosoma cruzi infections after 1 week of incubation. The aves refractoriness of the infections due to the innate immunity eliminates the T. cruzi inoculated into fertile eggs after 1 week of inoculation. The high sensitivity of the NGNA PCR, 10 femtograms, is 24-fold below the diploid T. cruzi total DNA, substantiates this information. The chicks that hatched from the T. cruzi inoculated eggs grew to adult life and died of heart failure. Those chickens had the kDNA mutations in the genome. The pathology showed cardiomegaly and severe myocarditis with the lysis of target cells. The chicks that hatched from T. cruzi inoculated eggs grew to adult life and died of heart failure. Those chickens kDNA mutations in the genome. The pathology showed cardiomegaly and severe myocarditis with lysis of the target heart cells. The scan shows the structure of the heterotransposon and the combination of primer sets used in the TP-tail PCR. The kDNA minicircle sequence inserted mainly averaged 70% in the chicken repeats 1, CR1, equivalent to the long interspersed nuclear elements 1, line 1 in humans. The kDNA mutations spread in all the chromosomes. Figure 27 shows the kDNA mutation and rupture of the PI3 kinase SMG1 gene at chromosome 14. The kDNA sequence is analog to the maxicircle uracilate guide RNA. The practical consequence of the kDNA mutations are occasionally seen in the chicken model of the Chagas heart disease. The insertion of the kDNA minicircle sequence in the code region of the dystrophin gene correlates with muscle weakness and heart failure. Some chickens with the kDNA mutated dystrophin gene showed cardiomegaly and foamy target cells with lymphocytic infiltrates. The inheritance of the kDNA minicircle sequence mutation, hitchhiking, and uncurbed biodiversity. The vertical transfer of the kDNA mutations, hitchhiking, and reshuffling observed in the chicken hatched from the cruzi infected eggs. The biodiversity showed by the crossings of the F0 chickens with the kDNA mutation into line 1 equivalent CR1. The F1 offspring hitchhiked kDNA mutation to the dystrophin gene. Further crossings reshuffled the dystrophin sequence into the kDNA, and the F3 progeny retained the kDNA CR1 sequence mutation. The integration of kDNA minicircle sequence into the genome of Trypanosoma cruzi infected mouse was observed after treatment with the leading trypanosoidal benzenidazole, ciprofloxacin, and AZT. The kDNA mutations were observed in the cruzi-infected mice treated with the leading trypanosomicidal benzenidazole, ciprofloxacin inhibitor of DNA gyrase and of topoisomerase II, and AZT inhibitor of reverse transcriptase. The treatment of cruzi-infected mice with those drugs did not inhibit the integration of the kDNA minicircle into the mice genome, and thus did not prevent the myocarditis. The chronic cruzi infections shift into the Chagas heart disease, usually not met in short-run studies. The overt chronic Chagas disease are seen in hospitals and specialized clinics. On one occasion, 3 heart disease patients were subjected to the kDNA PCR's clone and sequence. The table shows a minimum of 4 mutations, patient A, and of 8 mutations, patients B and C. The line 1 mutations were present at several loci of various chromosomes. In those patients, the arrays of kDNA mutations minimally documented possible genetically driven pathogenesis of Chagas disease. Further studies needed to unravel polygenic mutations at various chromosomes, blue ribbon breaking open clinical manifestations of Chagas disease. The inheritance of the Trypanosoma cruzi kDNA mutations is authenticated in the chicken model system refractory to the live infection. The chicks hatched from the T. cruzi infected eggs retain the kDNA sequence only. Assortments of mutations suggest the parasite-induced, genetically driven pathogenesis of the disease in the chicken model, and in some, 30% Chagas disease. Otherwise, the assortments of mutations at several loci and polygenic inheritance shall augment biodiversity in the great majority of the Trypanosoma cruzi infected population. A high-throughput platform required for the accurate diagnosis of the Trypanosoma cruzi infections. The emergence of Chagas disease requires health attention of skilled personnel. The etiology of idiopathic dilated cardiomyopathies could be unraveled by the nucleic acids tests, T. cruzi and DNA PCR. The development of new drug devoid of toxic effect is crucial to effective treatment and prevention of Chagas disease. A robust education, information, and communication program shall prevent outbreaks of T. cruzi infection worldwide. Thank you.